Hello everyone. This is Aashish Satyajith from Chennai Mathematical Institute, India, and today I'm going to be talking about optimal decoding of glycan information in single cells. To begin with, glycans are tree-shaped sugar chains that are covalently bonded to proteins in the plasma membrane. Here they engage in a variety of tasks such as encoding cellular identities and helping with cell-cell interactions and so on. Glycans are manufactured by an organelle of the cell called the Golgi apparatus, and so one can look at the whole process as the Golgi trying to convey some information to entities that surround a cell through these glycans. This raises a few natural questions. First of all, how do the entities that surround a cell decode the information that is presented to them by the Golgi apparatus using the glycans? And what are some limits on the information conveying capacities of the Golgi apparatus? To answer these questions, our approach is the following. Any perturbation to the Golgi apparatus will change the glycan distribution that you see on the cell surface. So you can view the Golgi as an information channel which takes as input a perturbation and gives a glycan distribution as the output. We measure the distribution of two types of glycans by measuring the levels of so-called lectins, which are glycan binding proteins, namely corne and WGA, that bind to glycans on the surfaces of the cells that we have. To make the perturbations, we use a reagent called Bruffeldin A or BFA, and what it does is it fragments the Golgi apparatus. So basically, the experimental setup is the following. We have a bunch of cells that we suspend in the solution. Then we add BFA and we let some time elapse. We have data points for both 30 and 90-minute time points. And then you label the cells with lectins, after which you do flow cytometry, and you'll get signal intensities that reflect the distributions of glycans that you have on the cell surface. And what we try to see here is to what extent can you infer the BFA concentrations from the lectin signals alone. To do that, we came up with a kinetic model which gives us predictions for signal intensities if you give it the BFA concentration and the time that has elapsed after adding the BFA. And this is obtained using least square square for 7 parameters using roughly 200 data points. Now let's look at the data. First of all, we see that the joint distribution of lectin values is very well approximated by a bivariate normal distribution. Secondly, we see that the lectin means show qualitatively different behaviors. In these plots, I've presented data for 30 and 90-minute time points for both WGA and corne. As is clear, in case of WGA, the signal intensities initially go up and then they come down, whereas in the case of corne, they just go all the way down. The fact that these two curves are of qualitatively different shapes seems to indicate that they convey different pieces of information and that it should indeed be possible to decode BFA concentrations given lectin signals alone. We approximate the covariance matrix of the bivariate distribution to be constant for a fixed time point, and this is a simplifying assumption that we have made. Now, to come back to the question of inferring the BFA concentrations given lectin signal values, we use Bayes' theorem, which gets us a decoder that can be represented as follows. In this diagram, for every pair of WGA and corne points, I've indicated what the most likely concentration is. And as one can see, there are two distinct regimes to the decoder, an all or nothing regime where you decode either to the maximum BFA concentration or to the minimum BFA concentration. And there's also an intermediate values regime where you do get values that are in between the max and the min concentration. Now, for the following plots, what I'll do is the following. I'll pick two values of WGA that are representative of both these regimes, and I look at the posterior distribution curves for different values of corne A just to see how the curves evolve. At, say, a WGA value of minus 0.75, which puts us in the all or nothing regime, we see that the posterior distribution curves are sort of V-shaped, and so you decode to either the maximum concentration value or the minimum concentration value, depending on whether the left end or the right end of the U or V is higher up. In case of the intermediate values regime, we see that there is the emergence of a peak and that the curves are actually an inverted V shape. And so this is why you get values of VFA that are in between the max and min. It is because the peak is in between this max and min, and the position of the peak will depend on the value of Corning. And just to be more explicit, what I have plotted in this figure is the concentration that corresponds to where the posterior curve is the highest. For example, in this case, for WGA Corning values of minus 0.7, we see that the posterior is highest at concentration 2, and so the diagram will have concentration 2 at that point. As one last point, we've also been able to estimate lower bounds on the communication capacities of the Golgi apparatus. To do this, we look at the mutual information between concentration and lectin signal values. Mutual information between two random variables will tell you how much information is contained in one variable about the other. And in our case, we estimate that the information that is conveyed by a single cell about the concentration is some 0.85 bits, or roughly 1 bit. So to summarize, we've come up with a model that gives some insights into how entities that surround a cell might decode the information that the Golgi conveys. And we've also been able to say statements of the form that the Golgi is capable of conveying at least this much information if it wants to. Thank you all for listening, and I hope to see you guys at the poster session.